We're going to continue looking at some vocabulary that we will be using frequently when we do punnett squares, and we've already talked about some of these vocabulary words, and I do believe that many of you may have already heard these in past science classes. So let's just kind of jump in and take a look at our first word, and it is called homozygous. Now, if you can remember prefixes of words, that'll really help you as you learn vocabulary in future science classes. So when you see the prefix homo, H-O-M-O, that means same. Homozygous is going to be referring to alleles that are identical to each other. And there are two different ways that the letters could be identical. We could have two capital letters. Okay, we already know that the capital represents a dominant allele. So we would call that homozygous because they're the same. And then we would put dominant on the end of it to represent the capital letter. We could also have two lowercase a's, which represent the recessive allele. So we would want to make sure that when we're writing out a definition that we know that homozygous is same, but recessive is tacked onto the end of that so that we can recognize the difference between two lowercase letters, which are recessive, versus two capital letters, which are dominant. Now, we also have the heterozygous word that you see here, and hetero as a prefix means different. So you noticed here that the letters are a capital with a lowercase. So heterozygous means that we have two different alleles, and whatever letter you choose. If it's A's, capital, lowercase, it's always going to be the capital and the lowercase of whatever letter we're using to represent the trait. We've already looked at phenotype and genotype. These may be words that you're familiar with already, but a phenotype is your traits. It's the organism's actual, could be their physical appearance, it could be things like on the inside, like whether or not you create the right type of red blood cell, or you have a certain genetic disorder or you don't. For Mendel, since he was again looking at those flowers and the pea plants, he had purple flowers versus white flowers. That was their trait, or he could have looked at tall or short. A genotype is looking at your genetic makeup. So this is, if I were to view someone, I can see outward traits. I cannot see genetically what that is made up of. So for instance, for the purple flowers or the white flowers, they're going to have a combination of DNA from their parents. Could have two capitals, could have one of each, could have the two lowercase letters. We're not going to be able to see someone's genotype just from their outward appearance. That's going to take a little bit of mapping out a family tree or a little bit more genetics involved. So here are some examples of how phenotypes and genotypes are linked up. A phenotype is always going to be very descriptive, maybe what color of eyes you have, brown eyes versus blue eyes. That would be a phenotype. So phenotype, physical trait, physical characteristic. A genotype is going to be what DNA letters do you have? Do you have the dominant allele for that trait? Do you have a recessive allele? Do you maybe have one of each? Brown eyes happens to be a dominant trait, so the capital B, whether you have one of them or two of them, give you brown eyes. Whereas for blue eyes, it's recessive, so you have to receive two recessive alleles, one from mom, one from dad, in order to have the blue eyes. And we're going to be looking at monohybrids in our practice problems. Mono means one, so a monohybrid is going to be a punnett square where we're looking at a single or just one characteristic. But before we get into practicing just one, we have one more law that Mendel kind of helped put together, and he did this by looking at two characteristics and how do the two characteristics match or pair together as they go through different generations. And he called this the law of independent assortment. And this just says that a pair of alleles, a pair meaning two, are going to segregate or separate independently, meaning that they don't have to be linked together. So for example, I think the picture would be easier for me to mark on here. He was looking at two traits in this parent. He was looking at the color of the seed, you see yellow versus green, and he was also looking at the seed, whether it was round or wrinkled. So he was looking at two traits at once, not just one trait. So you notice in the gametes that you have two different letters, a Y to represent that it was a yellow, and R to represent that it was a round seed. So you're looking at two traits. Now, as a parent, you know that you're only allowed to pass on half of your genetic information, which means that this original parent had two dominant traits for yellow, two dominant traits for the round, and they can only pass on one for color, one for the seed shape. So that's why we have just one Y and one R. If we look at the next generation then, the F1 generation, what would we get if those seeds were to cross-pollinate with each other? Well, you have two different possibilities here, and Mendel found out that this first one, the one with the big X through it, is not what mathematically he got. So originally it was thought that, for instance, a yellow seed will always be round, that those two characteristics were linked together. And so if that was the case, and you always had to have a yellow with a round seed, or a green seed with a wrinkled seed, then you'd only have two choices for each parent, and so your choices of offspring would be very different as well. You would never get any wrinkled yellow ones, because yellow would only ever pair with round. Well, Mendel did this experiment with pea plants, and what he found is that he got a wide variety of different pea plants when he looked at two traits together. He had some round and some yellow. He also had some green and round ones, no wrinkled, so he had green. And then here he also had some wrinkled combinations. He had wrinkled and yellow, and he also had some wrinkled and green. So that's where this law of independent assortment is. It just says that one trait is separate and independent from the other trait. They're not linked together. For instance, you can have a tall person with blue eyes. You can have a short person with blue eyes. They're not linked together and have to be seen together. All right, let's practice with some punnett squares. The first type of punnett square that we're going to be working with this week is called complete dominance. And complete dominance just means that there's two choices of phenotypes and that one allele is dominant over the other. So we're going to have a dominant trait and a recessive trait. For instance, in the purple versus white, you see that if you have the two capital letters or you have capital and lowercase, they still give you the same physical outward appearance of being purple flowered. So that's what complete dominance is. The capital letter, the dominant trait, is completely dominant over the recessive. And it does take two of the recessive traits in order for you to see the, two recessive alleles to see the white flower, the recessive trait. So we're going to do some examples here now with humans, and we're going to say that this is completely dominant, that we have two choices. We either have brown eyes or blue eyes. They tell us what our parents are to begin with. That's important. And they want to know what the phenotypes of each parent would be. They want us to do a punnett square. And then we need to write the expected phenotype and genotype ratios. Now I will tell you that as we do these, when I'm doing them here in the video, you need to also be doing them in your notes. And if you do not do them in your notes, then you're not going to get the points for having watched the videos if you don't do the work. So first of all, the phenotypes of the parents. The phenotype of this one, they have a capital B. That capital B means that it is completely dominant over the recessive, and so this parent is going to show brown eyes. And the little b parent, because the capital is not there at all, and they do have two of the lowercase letters, two recessive alleles, they're going to end up with the blue eyes. So there's the answer to the first part. We have the phenotypes of each parent. Now to do a punnett square, we know that this parent here can only pass on half of their DNA to their offspring. So they can't pass on the capital B and the lowercase b. They can only pass on one or the other. So we're going to take their alleles and we are going to segregate or separate them. This is what happens during meiosis when the homologous chromosomes are being, separated. The one cell could get a capital B, so this is one gamete. The other gamete has a little b possibility. Now for the other parent, since they have little b twice, every gamete that they create, and that cell just represents a gamete. You do not have to draw a circle around all of your letters. Every gamete that the blue-eyed parent creates will have the recessive allele for blue eyes. Now to finish out your boxes, these 4 empty boxes, all of these letters get written across. You're going to put that little b in every box. The capital B you are going to be going down with. So where these two meet, you're going to have a capital B and a lowercase b. Then in this box, you're going to have the little b coming down and the little b coming over, so little b, little b. In this bottom left-hand box, we have the capital B coming down with the little b coming over. And in the final box, we have the 2 little b's. So this would be the Punnett square. Now these boxes represent the zygotes. This would be what the offspring, or the child, would look like if those 2 gametes were to come together and form new life. We want to know what the expected phenotype and genotype ratios are. Well, first of all, for phenotypes, our choices are brown eyes or blue eyes. Those are the only 2 physical characteristics that they've given us. So we have brown or blue. And if you take a look at your Punnett square, this offspring has a capital B, and this offspring has a capital B, and these 2 over here do not. So the only 2 that could have brown eyes were these first front 2 boxes. So that is brown eyes is 2/4, or you could put it at 50%. And then for blue eyes, you have these 2 boxes. The little b combinations have no capital letter, so that's 2/4 or 50%. I'll normally stick with the percents, but just to, some of you may want to do fractions, and that's okay. Now for genotypes, genotypes are the DNA letters. So in this case, we could have big B, big B, big B, little b, or 2 little b's. Now if you look at your 4 boxes, big B is not a possibility at all. There's 0% chance of getting that. For little b, that happens once and twice. There are 2 boxes out of our 4 that give you that. And little b is also 2 boxes out of 4, or 50%. So that would be the phenotype ratios and the genotype ratios, and we have answered all of the questions that they've asked of us. So let's go on to question number 2. Now this one we're gonna get started together here. Homozygous recessive. So go back to our vocabulary. You may have to flip a page in your notes. Homozygous means same. So the letters have to be identical, and recessive means the lowercase letter. So the genotype of your parent here is going to be little b for the homozygous recessive. And they also asked for the genotype and the phenotype of the parent. So we know little b has to be the blue eyes because there's no capital B there. Our other parent is heterozygous. Heterozygous means different. So we have 1 of each. And because of the capital letter there, we know that they have brown eyes. So we've completed question 1 there. And now we need to do a Punnett square. So for our Punnett square, we have our little b parent. And it doesn't matter, you could put the little b's on the side and the capital little b on the top. It doesn't matter. But just going to keep them in the order that I wrote them down in. So there's our Punnett square. We've segregated our alleles because the parent can only pass on 1 of the 2. And I'm going to go ahead and fill out my boxes by bringing my letters over and down. So there's my Punnett square completed. Now the question is, what are the chances that these 2 parents would have a child with blue eyes? So we need to look at our 4 choices because these are the children possibilities. This one right here represents blue eyes, and this one. So we have a 2/4 or 50% chance of blue eyes. And then it also says, what are the chances that they have a heterozygous child? So we need to know the vocabulary. Heterozygous is different, and that would be the capital B, capital B, and that happens 2 out of 4, or 50%. So we're going to be working through many different types of Punnett squares. This is the first one. This is complete dominance. They either have brown eyes or they have blue eyes. No other trait in the middle, no other third choice.